been a true pleasure to interact with members of CBTN, Brian, Adam, and I'd like to talk to you a little bit about profiling, its application to childhood brain tumor samples. So I think most of you know that pathologists typically would use the microscope as their primary diagnosis. And that was really how we've been doing it for over 100 years. And that works many times. Sometimes it doesn't give the information we need to make an accurate and precise diagnosis. There are some cases that are just difficult, particularly pediatric brain tumor treatment, where interpretation is subjective, ambiguous cases, and we often have to give a descriptive diagnosis sometimes. So, and that's reflected in the WHO, which here's an example of some of the entities from 2007, where everything was named how it looked on the microscope, essentially, where molecular aberrations were beginning to be known, but they weren't emphasized in the diagnostic criteria or the definition. But here's some examples of entities in the most recent WHO book, where many entities underlined in red are defined by their signature molecular alterations. And so we're beginning that process of incorporating molecular alterations, and I think that process will continue. And one way we can do that is to incorporate, I think, methylation profiling into a diagnostic criteria, because that provides new information that isn't present in other kinds of omics technologies. I think probably most of you are aware of this work from Heidelberg, where a number of samples that had been given a diagnosis of CNS-PNET were subject to methylation profiling. Many of these tumors sorted out into known entities, glioblastoma, ependymoma, ATRT, et cetera. And some of them did not, and 4 new entities were described here. I won't go over them in detail. It's likely that most of you know this already, but that many of these CNS-PNETs really could be more precisely classified according to methylation. And the relevance here is not just academic. And here's a study conducted by some of you who are, I'm sure, in the audience, where you went back and looked at samples from a clinical trial on CNS-PNETs and found out that a proportion of these, in fact, the majority of the non-pineal region tumors were actually would not have been included in the intent for this trial, really underlining the importance of using something like methylation profiling to get to an accurate diagnosis. So one example of how it's changed our classification is really shown here in these 3 histologies. In 2007, these were 3 different entities, etanter, ependymoblastoma, and medulloepithelioma, based on how they looked on the microscope. And you can see that they each look different and distinct. However, it was found that using methylation, one could not distinguish them. They had essentially identical epigenetic changes, and so that one could not tell one from each other based on the DNA methylation profiling. And consistent with that is the genomic aberrations were similar too. Here in these examples of 3 cases, etanter, ependymoblastoma, medulloepithelioma, each of them has gain of chromosome 2, and most importantly, each has amplification of the C9NMC locus. So this is an example of convergence of diverse histologies into a single class, as opposed to a divergence, which we talked about for the PNETs, where the similar kind of PNET histology diverged into different methylation classes. With respect to ETMR, that was a convergence of different histologies into a single class, really, I think, underlying the interaction between histology and epigenomics in classification. And then, of course, another use of methylation is really subtyping of known entities, for example, medulloblastoma or ependymoma, et cetera. So many of you are probably aware of this figure from this paper, from the paper from Heidelberg several years ago. This is a t-SNE plot of a variety of CNS tumor entities. In this example, there are 82 different tumor entities distinguished by methylation. A classifier was developed, and that's being used more and more classify tumors on a clinical basis, and we're doing that here. A second generation Heidelberg classifier has been developed and has increased number of classes and subtypes. And here at the NCI, we're using an alternative machine learning method to develop a complementary classification, not to replace the Heidelberg classifier, but really to complement it. What we find in methylation is that not all tumors classify with a high confidence score. Probably about 2/3 or thereabouts cases that are sent to us for methylation actually do classify with a high confidence score. We find that having complementary classifiers can help provide that consistency to accommodate when we don't get a high confidence score for a single classifier. And so what is the value here? Besides showing nice pictures, nice Tisney plots, et cetera, what is the actual value of methylation to get to a more precise diagnosis? So to look at that, we retrospectively looked at the cases that we've been profiling here, where each case sent to us was run by methylation, and to ask the question, really, how much does it contribute to the diagnosis? Does it simply confirm it? In which case, we're not really adding much value except for confirmation. What about subtyping? We've talked about subtyping of medulloblastomas, ependymoma, et cetera. But more importantly, how often can we resolve a diagnosis that was not previously identified using state-of-the-art techniques, histology and NGS, et cetera? So we looked at about 1,000 cases over consecutive cases over a 2-year period, and compared the pre-methylation or the pre-classifier diagnosis to the post-classifier diagnosis, and looked at these various outcomes. And the results are summarized here. I won't go through all of the details of this busy slide, but the bottom line is of the 1,000 or so cases, about two-thirds of them got a classifier score above the confidence score cutoff of 0.84. And of those, about a third, 35% were confirmed. That is, the methylation didn't change the diagnosis. It confirmed it, which was helpful, but it didn't change it. 13% the diagnosis was consistent, but allowed more refinement, medulloblastoma subtyping, et cetera. But in 18% of the cases, these were the cases where methylation had a substantial impact, where the classifier provided information for a diagnosis that was not suspected or defined before methylation. And some of those pictorial examples of how that worked is shown here. I won't go into a lot of detail of these Sankey plots. Essentially, on the left side is the pre-methylation diagnosis, in the middle is the methylation classifier, and on the right is the final diagnosis. I'll just point out that for one diagnosis that is relatively common in neuropathology, that of glioblastoma, even here, methylation has a role, where most of the time the diagnosis was confirmed as IDH wild-type glioblastoma. However, there were a proportion of cases where the methylation classifier gave us a different picture, something that was alternative to glioblastoma. And they're shown here on the right. Some of these changes, although rare, had clinical implications with respect to WHO grading. So even on a tumor type that is relatively straightforward, or thought to be relatively straightforward in neuropathology, methylation can have a role in some cases. Here on cases that we received without a diagnosis, where they were considered unclassifiable. Methylation did not resolve all of them, but about half of them. 144 out of 289 cases were resolved by methylation. I will say that these were enriched for childhood brain tumors, where we find that they can be more difficult to classify using routine techniques, and where methylation is particularly helpful. Again, not in every case, but in a good proportion of the cases, methylation could resolve it into a final integrated diagnosis. So this is just summarizes that here, where it does have an impact on a substantial number of cases. I will point out that 1/3 of our cases did not receive a high confidence match, so there is still room for improvement in methylation methylation-based classification. There's an opportunity to improve or complement the Heidelberg classifier to enable some kind of ability to get some kind of diagnosis on these cases that do not give a high confidence score. We can often use orthogonal data, such as DNA copy number signatures, fusions, et cetera, when applicable, to help us resolve a diagnosis when the classifier score isn't high. So there is, I think, room for improvement in methylation-based classification. Another opportunity in methylation-based classification is identifying new entities. I'll just go through one of them that we've described recently, and that is of a posterior fossa ependymoma rare subtype that we found was different from the classic PFA, PFB subtypes that were previously described. Basically, what happened was we identified several ependymomas from the posterior fossa that didn't match to either PFA or PFB. They clustered a little bit outside of those two entities. And then we found that several of them had a mutation in the gene ACVR1. And I think most of you know, ependymomas have been classically separated into these various groups from the work of Richard Gilbertson and others. And what we found was this particular rare subtype didn't fit into that. It didn't fit into the PFA or the PFB. And here's a representation on a UMAP showing these tumors that were posterior fossa ependymomas. They were neither PFB nor PFA. But we found that they had a mutation in ACVR1, a gene, as you know, mutated with some frequency in DIPG. So this gives us an opportunity to identify potentially new subtypes of tumors that weren't previously recognized based on their methylation classes. This kind of thing, I think, has been looked at by others. Here's one paper looking at pediatric series of pediatric brain tumors. We're here, similar to what we showed, a number of cases where the pre-methylation diagnosis based on the methylation class could be revised. So I think here's just some other examples there, where I think there is, I think, a good rationale to consider methylation profiling, especially in rare or difficult to diagnose tumors, especially those of children. With respect to CBTN in particular, it's been quite a rewarding experience. We've begun to look at this. We've profiled over 1,700 CBTN samples to date and are still analyzing the data. Here's just some of the more frequent classes that we found: posterior fossa, pilocytic astrocytomas, a variety of ependymoma subtypes, a variety of pediatric type high-grade gliomas, grade 4 tumors that were previously discussed at a previous talk, et cetera. This data is just emerging, and we're beginning to mine that data, but I think it will complement some of the other omics data in CBTN to really help us understand these tumors and help nail down a diagnosis when that diagnosis is ambiguous. So just to summarize, methylation profiling result isn't necessarily a diagnosis on its own, but I think is an important and complementary tool alongside other information to arrive at an accurate diagnosis. And I think where it really works is when you have integration with other omics technologies, with the histology. That's, I think, where the special sauce, where we really get into precise and accurate diagnostics. And I think best practices is going to be for diagnosis, especially for childhood tumors, that methylation profiling is going to be of great value to get to the right diagnosis in these cases. So with that, I will stop and thank you for your attention.